We'll be talking about the alternative complement pathway in this video. There are actually two different parts to this pathway. There's what we call the fluid phase C3 convertase and the membrane-bound C3 convertase. So I'm going to go through both of those different types of this alternative pathway with you first. So the fluid phase C3 convertase is going to result in generation of a C3 convertase that's going to remain in the blood. And the main role of this fluid C3 convertase is to make more C3b that can go on and be used to bind to the membranes of pathogens. Remember, C3b is an opsonin. So this fluid phase is really just to produce a lot of C3b in the blood that can circulate and go and bind membrane pathogens, or membrane of pathogens. So we're going to start with our C3 molecule. So here's C3, and kind of the first step or thing to recognize in this pathway is that C3 is always undergoing a spontaneous hydrolysis, and it forms C3H2O. And that's going to be this molecule in our model, this C3H2O. Now what happens next is C3H2O is going to bind a protein called factor B, which is shown here. So we result with C3H2O plus factor B. The next step is that this complex is going to bind another protein called factor D, which is shown in pink. And then what happens is that factor D actually acts upon factor B and causes it to be cleaved. We're going to result with two small molecules then. We'll have factor BB, or capital B little b, and then we'll have factor BA, which I'll just put over here because it's not really used in this pathway. So we're left with this complex. D actually dissociates, so we'll erase that. Just put that off to the side. But we're left with this complex of factors C3H2O and BB. So this right here is C3H2O, the blue molecule, and capital B little b, which is our red molecule that's been cleaved and remains behind after factor D had acted on factor B. This right here then, this blue protein with this red protein, is what we would call a fluid phase C3 convertase. So exactly what we wrote up here. So this C3 convertase is going to be responsible for taking more molecules of C3, which would be shown right here. So that C3 convertase can act on C3 and basically generate our C3b and our C3a. So C3a would be the little molecule or protein. C3b is the larger protein. Okay, so that is what happens in the fluid phase of this alternative pathway. We're generating this C3 convertase that's going to remain in the blood, and its main function is to generate more C3b, or that opsonin. We sometimes call it the alternative tick-over pathway because of the spontaneous hydrolysis of C3 and the fact that it's constantly being replenished, and we're always making more C3b. You can think of it as like a natural turnover process in the cell. Now, the other part of this pathway is what we call the membrane-bound C3 convertase. So I'm going to write that up here. And this relies on the formation of C3b. So that can be from the fluid phase C3 convertase, as we saw over here. That's going to generate C3b from the C3 molecule. I can put that back in there. C3 going to C3b and C3a. So once we have C3b, In this membrane-bound C3 convertase version of the pathway, that C3b is going to associate with the membrane of our pathogen, such as our bacterial cell. Now what's going to happen, again, we said this is the membrane-bound version of the pathway, so everything is going to start to assemble at the pathogen membrane. So we're next going to have factor B associate with C3b there. So here's factor B, our red protein. And then we're going to have factor D associate with that complex. So right now this complex is often abbreviated C3 little b capital B. That's factor B associated with C3b. Now we have factor D come in. And D has proteolytic activity, so it's going to cleave factor B into the two smaller subunits. One of those will be BA, which I'll just put up here again by that label. And then the other one would be the protein capital B, little b. And then factor D is done with that cleavage, so we're going to remove that and just put it up here. So what we'd be left with after the cleavage process that factor D caused is we still have C3b, and now we have the smaller subunit of factor B, which is capital B, little b, to distinguish that it's been cleaved. So this complex here, C3b, BB, is what we call our membrane-bound C3 convertase. Okay, so it's going to act upon C3, and it's going to generate a lot more of our C3a and our C3b. So the next step is to form what we call the C5 convertase. So one of the ways in which we can form that C5 convertase is through a protein called properdin. Now properdin is something that's going to help stabilize this membrane-bound C3 convertase and really make sure that it stays in the membrane. So properdin comes along and binds to that. And it helps it be stabilized so that now it can actually start to form C5 convertase. So how C5 convertase is formed is we just take this C3 convertase and we add another copy of the complement protein C3b. So if we were to kind of abbreviate this in the typical nomenclature for the complement pathway, we'd have C3b. Factor B here is the larger subunit of the cleaved version, so it's capital B, little b, and then we just added another molecule of C3b. So this whole complex now is what we would call our membrane-bound C5 convertase. So C3b with capital B, little b, or that cleaved factor B, plus this another molecule of C3b, all stabilized by properdin. These three subunits or proteins are what make up our C5 convertase. So from there is where this is going to now converge with the rest of the pathways. They all work the same way at the end. So we have a C5 convertase. Running out of room here, but basically that's going to act upon C5. We're going to generate C5a, which is one of those inflammatory mediators. And then we're left with C5b at the membrane. Okay, and then once we have C5b, I'm just going to move that over here in a different region of the membrane so we have space still. Once we have that generated, then we have C6, C7, C8, and somewhere between 10 to 19 copies of C9 that are going to come in. And this whole complex then is what we call our membrane attack complex. So really everything that happens after the generation of the C5 convertase is the same in the alternative pathway. The main difference is how it gets started. We have this fluid phase convertase, which is formed based on spontaneous hydrolysis of C3. into this C3H2O form, and then that associating with factor B and factor D to make our fluid phase C3 convertase. That can then make more C3A and C3B. The C3B can come over here and be a membrane-bound version, which associates with factor B and D. The cleaved factor B version binds to C3B to form the C3 convertase that's membrane-bound, and then that associates with properidin and another molecule of C3B to form the membrane-bound C5 convertase, which then is responsible for initiating the membrane attack complex formation. So this pathway is a little bit complicated in that we're looking at sort of two arms of it, but you'll see again that many of the same principles of the other complement cascades apply here in the alternative pathway. Main thing to think about is that there is the blood version, the fluid phase, and the membrane-bound version in this pathway. You can read in your textbook more about how this pathway has been discovered, specifically how properidin was discovered and the classical experiments that they did to identify that protein and its role in this membrane-bound alternative pathway. So now would be a good time to complete question 3 on the Google worksheet or the complement activity. And once again, if you have any questions about alternative pathway, please feel free to reach out to me.